Hey everyone, so this is where we left off on our last round of notes, just talking about apoptosis and how programmed cell death is a normal function of cells. So if we kind of put everything into perspective and just kind of flash back here in terms of talking about the cell cycle in general, um, we we emphasize that the cell cycle is this normal process in which the cell goes through growth, manufacturing proteins, making more organelles, carrying out the functions of what that cell is designed to do. At some point in time, that cell goes through DNA replication during S phase, re-enters another period of growth and function in preparation for mitosis and cell division. And once that mitosis and cell division is carried out, cell enters back into this normal period of interphase where growth, manufacturing of organelles, proteins, carrying out its functions, etc. And this is just a regular pattern in the cell cycle. Now, something may occur within that cell cycle that leads it to that programmed cell death. Could be internal conditions within the cell, external conditions outside of the cell. But again, it's all part of the normal cell cycle of life and death of cells. Um, except in such circumstances when it's not normal. And that is essentially the role that cancer plays within the cell cycle. And that's exactly what cancer is a disease associated with uncontrolled cell division. All right. Um, okay, but what we need to understand there is it is uncontrolled cell division of abnormal cells. All right. And so, what makes these cells abnormal in the first place? Well, recall that the cell cycle is controlled by checkpoints. And what we need to remember is that these checkpoints are proteins. All right. And they determine whether or not a cell advances to the next stage of the cell cycle and undergoes division or not. And cancer essentially arises in one of two ways. One way is mutations, all right, mutations in the DNA. So remember that the DNA is the genetic code, right? DNA is the code for proteins, and that includes all proteins, even those checkpoint proteins. And so if there are mutations specifically in the genes coding for those checkpoint proteins, these mutations can lead to a failure in the checkpoints. All right. So that's one way. Um, and another way is a normal cell. So it's a completely normal cell, but embedded within a benign tumor, right? And remember, a benign tumor, if you started to do some of your investigation with cancer, and I think we kind of all have heard that before at some point in time, a benign tumor versus a malignant tumor, and we'll talk more about those. But a benign tumor could be like a mole on your skin, right? It's virtually kind of harmless. But there can also be tumors, benign tumors, in which they surround a normal functioning cell. And what happens is that cell can be starved of oxygen. Cancer cells are very good at robbing other cells of their oxygen supply. And so as a result of this lack of oxygen, um, the cell switches to anaerobic respiration, which should make sense, right? We know that when there isn't sufficient oxygen um, around that our cells will undergo anaerobic respiration. But an extended period of anaerobic respiration in a cell can ultimately cause the cell's self-destruction mechanism, that apoptosis, to be turned off. The cell then becomes immortal in a sense, and it will continue to divide, and a tumor will grow. Now, that tumor could still remain benign, right? And if you watch this video, which I do recommend, do a YouTube search on Cancer 101 National Geographic, and it'll emphasize that difference between a benign tumor, which is localized and kind of stays put, versus malignant tumors, in which as the cells grow and divide, those cancerous cells invade surrounding tissue. They can even invade the bloodstream or the lymphatic system, and then these cells can be transported to various other parts in the body, and then you have the spread of cancer, right? Well, that's where cancer gets its name from, right? Like the cancer term for crab, right? Which is spreading, right? The spreading legs of a crab. Now, if we look down here on the bottom right-hand side, what I want you to understand, or some terminology that you should know, is these genes that code for these proteins that regulate the cell cycle. These genes are called proto-oncogenes, all right? And proto-oncogenes are basically normal genes coding for checkpoint proteins, all right? They're referred to as proto-oncogenes. Now, mutation, as it shows here, mutations in those DNA repair genes, or sorry, mutations in the proto-oncogenes that control, that produce checkpoint proteins and control cell division, can lead to what we call oncogenes. And oncogenes are essentially cancer genes. All right? And so hence the term, the study of cancer is called oncology, right? And so you get these oncogenes, and this leads to abnormal cell division, because you no longer have normal proteins, normal genes that produce normal proteins that regulate cell division. And so essentially what you have is now uncontrolled growth, right? Uncontrolled cell division due to mutations in proto-oncogenes. All right. And if we look here, this really defines some major differences between normal cells and cancer cells. Typically cancer cells have enlarged nuclei. They can even have multiple nuclei. But when these multiple nuclei and enlarged or multiple and enlarged nucleoli, they tend to reduce the cytoplasm, which would reduce function of the cell. Remember, many processes occur in the cytoplasm of the cell, so that becomes reduced. They have small cytoplasm, multiple nuclei. Their cells tend to be very irregularly shaped. Sometimes even enlarged cells, or definitely enlarged nuclei, and essentially lose normal function, right? And cancer results. So the video here looks specifically at how normal cells have many repair mechanisms. So there's lots of things cells have to prevent this from happening, even when things go wrong. So a normal P53, and this is just the name of a particular gene that codes for a protein, and let's say a cell was damage was done to DNA of a cell, and this causes cell cycle abnormalities, this hypoxia, right, or lack of oxygen, right, lack of O2. And what will happen is this P53 gene will kick in, producing proteins that will lead to a cell cycle arrest. So basically, hey, things aren't going right, let's stop the cell cycle. And one of two things will occur. Either it will trigger apoptosis, programmed cell death, which is a good thing, and prevents that cell from dividing and producing more messed up cells. Or in the case of this P53, there can be DNA repair. So whatever that damage was that caused the problem in the first place, and a cell cycle restart. And that's a normal functioning of many of our cells, and that's why we can stave off cancer. Unfortunately, in the case of a mutated P53, you get DNA damage, cell cycle abnormalities. So here's our messed up P53. The cell cycle doesn't stop, it continues, and cells become cancerous, and we end up with this scenario over here. Okay, so mutations to proto-oncogenes, genes controlling cell cycle, checkpoint proteins, and that leads to our tumors enlarging and complications. We can imagine what happens when cells of particular tissues start to lose their functions. Well, then we understand organs don't work properly, potentially organs start to shut down, and we're in trouble. All right, that's the end, a short one. I plan to post video number 4 on meiosis tomorrow. Um, and if you have any questions at all, don't hesitate to send me an email for tomorrow. I will be online and responding to things. Otherwise, enjoy the rest of your time off, and we'll see you Wednesday.